Hello, my dear students. So I'm Professor Sakti Vayapuri. So my next lecture in PM1A is going to be on lipids and lipoproteins. This is quite important lecture because we're going to study quite a lot about lipids and essential information about lipoproteins. And they are really important throughout your M.Pharm program and our life as well, because fats or lipids are important components in our body, and any dysfunction of fats and lipoproteins obviously will lead to different cardiovascular diseases that you're going to study in more detail in next year. Okay, let's get on with this lecture. So what are the learning outcomes for this lecture? So after this lecture and further reading, you should be able to explain the importance of lipids in our body. And you should be able to understand the different types of lipids and their biological significance in our cells or body. And then you should be able to define lipoproteins and their architecture. And then you should be able to demonstrate the functions of different types of lipoproteins in detail and illustrate the significance of tightly controlled regulation of lipid transport in the body. Why the lipid transport has to be very tightly controlled, and if anything goes wrong in this lipid transport, how this is going to lead into cardiovascular diseases that we will touch on next year. So overall, you should be able to understand the importance of lipids for cellular energy requirement and the lipoproteins in transporting these lipids. So that's what we're going to focus mainly on this lecture. And because you are on part 1 and this is basic lecture, you're going to learn only the basics about lipids and the lipoproteins. So the more details you're going to learn about lipids in lipid metabolism lecture, which is coming in again the next 2 weeks, and then about cardiovascular diseases in more detail you will study next year. Okay, so what are lipids? So lipids are commonly known as fats. So many people call it as a fat in common term, and scientifically we try to call it as lipids. So in Greek, lipos means fat. Okay, so what is it actually? It's organic molecules. It's a macromolecule. Similar to carbohydrates and proteins in our body, we do have lipids as another macromolecule, macrobiomolecule in our body, and it's really essential to maintain our body and to maintain the functions of our body. So similar to carbohydrates and proteins, lipids are made up of carbon, hydrogen, oxygen, and sometimes lipids will have phosphorus group, okay, or phosphate group. So these are mainly made up of carbon, hydrogen, oxygen molecules. So lipids are mostly hydrophobic molecules, which means they repel water or insoluble in water. Okay, so if you take a drop of oil and put it on the water, you will see the oil is floating, not mixing with the water. So what are the main functions of lipid? Why do we need lipids in our body? So lipids act as important energy storage in our body. So whenever we have excess amount of sugars in our body, for example, glucose and other sugar molecules, it will be stored in the form of glycogen, very small amount, and then the rest will be converted into lipids like fatty acids. We're going to talk about that in more detail soon. And that's how it will be stored, because lipid storage is the main energy storage in our body. That's what is going to really give us more energy when we are starving. And lipids are really important to make up our cell membranes, because as we all know, our cell membranes are plasma membranes, and it's basically phospholipid bilayer membrane. So it's made up of lipids. So every single cell contains cell membrane, and that cell membrane needs lipids to make. And the lipids provide thermoinsulation for our body, and that's what is really important to regulate the heat in our body. So under the skin, we got like thick layers of lipid, and these layers of lipids are really important to maintain our body temperature of 37 degrees Celsius. Without lipids, it will be hard to maintain the temperature. And the lipids are really important to synthesize several hormones, particularly steroid hormones. They are really essential to regulate our body function, and lots of secondary messengers and other biologically important molecules. For example, bile acid, that's really important for the digestion of lipids in the gut, and some vitamins, for example, vitamin D. So these are all important molecules and important functions in our body, and without lipids, these functions and these activities cannot be carried out. Okay, what are the major classes of lipids found in our body? So there are 5 major classes of lipids. Within these classes, there will be small classes, but we're not going to touch on that in too much detail. And there could be more minor classifications, which again we are not going to touch on. But most of the lipids in our body can be broadly classified within these 5 classes. So what are these classes? So the first one is called fatty acids. Most of the fats in our body will be in the form of fatty acids. And then the second one will be triacyl glycerols or triacylglycerides or triglycerides. The third type of lipid classes are glycerophospholipids. The fourth one are sphingolipids, and the final one is steroid. Okay, so these are the 5 important classes of lipids: fatty acids, triacylglycerols, glycerophospholipids, sphingolipids, and steroids. So we're going to talk about each one of these in a little bit more in detail. So the first one, we're going to talk about fatty acids. So as I said before, most of the lipids in our body are present in the form of fatty acids. Okay, so what are fatty acids? So free fatty acids rarely occur in nature. Okay, mostly they will be bound to other molecules. So for example, they are mostly esterified. Okay, so they will react with an alcohol group and make esters, and they will be present in esterified form. So the free fatty acids are really rare in nature in our body. Only during metabolisms or digestion you will see free fatty acids. Rest of the time during storage and transport they are likely to be esterified, and they form monomer like structure in most lipids. Okay, fatty acids will form monomer like structures in most of the lipids, and without these fatty acids those lipids cannot be formed. So this is how the structure will look like. So the fatty acids will have a long hydrocarbon chain, or you can call it as a tail, hydrocarbon tail. Okay, so you can look here, all of these like a long aliphatic chain, hydrocarbon chain, it will act as a tail. And then it does have a carboxyl group, and this carboxyl group acts as a head. You can see here carboxyl group right at the end, and that acts as a head. So that's why it's called fatty acid. Okay, so it's got carboxylic acid group right at the head, and then it's got aliphatic hydrocarbon chain. Okay, so this is why it's called fatty acids. The predominant fatty acids in our body are present in the form of C16 or C18, which means it will have either 16 carbons or 18 carbons. This is how most of the fatty acids will be found in the body. So try to remember this structure. So it does have a hydrocarbon aliphatic chain tail. Okay, but it does have a head group with the carboxyl group. So that's why they are called fatty acid. Okay, so fatty acids are generally classified into 2 forms, and you will come across this name quite often because of the modern world, because people are talking about saturated and unsaturated fatty acids all the time. So they are broadly classified into saturated fatty acids and unsaturated fatty acids. So what are saturated fatty acids? So saturated fatty acid means they will not have any double bonds except in the carboxylic group. Okay, so if we look at the aliphatic chain or hydrocarbon chain, there will not be any double bond, which means all the carbon valencies are is fulfilled. So they bound to hydrogen molecule in order to fulfill all the valencies. So you will not see any double bonds in the chain, in the hydrocarbon chain. So these are called saturated because all the carbon valencies are saturated. So normally this type of fatty acids will be in the form of solid at room temperature for around 20 degrees Celsius. It will be solid. The best example for you is butter. So we all use butter, and the butter is mostly in solid form, even if you leave it in room temperature. And the butter, mostly from animal sources, will have full of or plenty of saturated fatty acids. Okay, so that's why we keep saying, or people are keep saying, that the animal fats are not good for health. Plant fats are really good for health. So because these saturated fats can increase the LDL level or LDL cholesterol level, and elevated level of LDLs are not good for health because they can lead into cardiovascular diseases. So saturated fatty acids means there will not be any double bond in the hydrocarbon chain. and they are mostly from animal origin, like for example butter using animal milk, and these are not good for health because they can elevate the LDL levels and subsequently lead to cardiovascular diseases. Okay, what are next classes of fatty acids? So the next ones are unsaturated fatty acids. Okay, what do they mean, unsaturated fatty acids? Which means the hydrocarbon chain will have double bond. Okay, because these carbons are not fully saturated and therefore they will have a double bond. So these fatty acids will be in the form of liquid at room temperature because they don't pack very well. So if we put all these molecules, the saturated fatty acids, in a small container or a small space, it will get packed really very well because they don't have any crinkle or bent like structures. But if we look at the saturated unsaturated fatty acids, they do have this type of bent structures, which means they don't pack very well. So for example, in a small space, for example, 1 centimeter, 1 centimeter, if you put 100 saturated fatty acids, you may only fit 40 to 50 unsaturated fatty acids in this place because they have bent structure or crinkle structures. Therefore, you will not be able to pack them really very well. So that's the main difference between saturated fatty acids and unsaturated fatty acids. So saturated fatty acids can be stored in large quantity within a small space, but unfortunately, unsaturated fatty acids cannot be stored in that much quantity in small space. So the best example here is oil, and most of the plant fats will be in the form of unsaturated fatty acids. And that's why people will say, or cardiovascular specialists will say, reduce or cut down your animal fat and increase the amount of fat you are taking from plants in the form of oils. Again, within the unsaturated fatty acid, there are different classes. For example, mono unsaturated fatty acids and polyunsaturated fatty acids. Mostly plants do have most of the fatty acids in the form of polyunsaturated fatty acids, which means they will have multiple double bonds in their hydrocarbon chain. Okay, so if it's having only one double bond, it's called mono unsaturated fatty acid, and then if it has multiple double bonds in the hydrocarbon chain, it's called polyunsaturated fatty acids. Okay, so they have several disulfide bonds, and polyunsaturated fatty acids are really good for health, and that's why doctors recommend to take plant oils in most cases and cut down animal fats. Okay, so why this numbering? So you will see this numbering for fatty acids all the time. Sometimes they will say delta 9, delta 12, octodecatonic acid. Sometimes they will say omega fatty acids, 18:2 omega fatty acids. So it's really important to learn a little bit about this numbering system. So for fatty acids, the numbering can either start from carboxy terminal. Okay, so if it's starting from here, like a carboxyl group, number 1, the carbon is number 1, and then it's going towards the methyl group, methyl end, then it's called delta numbering. Okay, so the numbering, if it starts from carboxyl group and goes all the way up to the end of methyl end, it's called the delta numbering. But in some cases, the numbering will start from the other way around. It will start from methyl end and then it will go towards the carboxy terminal. Okay, so that's what is called omega numbering. Okay, for example, here omega 3 and 6 are important classes of polyunsaturated fatty acids. Okay, so if we got the double bond at omega carbon 3 or 6, they are really good polyunsaturated fatty acids. So here you got an example that's called a linoleic acid generally. You can call this as a delta 9, delta 12 octodecatonic acid, because if you call the number from carboxy terminal, the double bond is present in the 9th carbon. You need to take the first carbon, not the next one. OK, so 9th carbon and then 12th carbon. That's why it's called cis. Cis means position. I will come to this in a minute. So delta 9, delta 12 octodecatonic acid. That's the name for linoleic acid. The same fatty acid, same linoleic acid can be named as 18:2 omega 6 fatty acid, which means it's got 18 carbons. That's why this 18, and then 2 means it's got 2 double bonds. Okay, and usually for the name they will use only the first double bond position. So here from this end, this is a sixth carbon and the double bond is there in the sixth carbon. Therefore, it can be called as 18:2 omega-6 polyunsaturated fatty acid. So this is the numbering. So the delta numbering will start from carboxy terminal, omega numbering will start from methyl end. Okay, what's cis? So cis means this double bond and the hydrogen subsequent hydrogen group will be on one side. Okay, this is what will provide that crinkle like or bent like structure for that fatty acid. So you can see here clearly both the hydrogen here on one side. Okay, it's not in the opposite. This is what is called cis position. So the hydrogen will be in the cis position in the same orientation, and therefore it will nicely bend the fatty acid. However, if you look at the trans fatty acid, the hydrogen position is in opposite direction. So when the hydrogen position, even though it's got a double bond, when the hydrogen position goes in the opposite direction, then this will not be bent anymore. Okay, so the best example is artificial hydrogenation of some plant fatty acids, plant oils. For example, it's called vanaspati. It's a plant oil, but artificially hydrogenated. So that will make this type of trans double bonds, and that will look similar to saturated fatty acids. Therefore, this is no good. So, okay, because this is looking exactly similar to saturated fatty acids, and therefore they can pack really very well within small space, and therefore it's not good for health. So we need to take naturally available cis fatty acids from plant sources, and that's what is really good for our health. Okay, so next we are going to talk about triacyl glycerols. They are also known as neutral fats because they will not have any acidic group because it will be neutralized, and they are also called triglycerides. Okay, so most fats and oils occur as mixtures of triacyl glycerols. Okay, as I said before, the freely available fatty acids in our body are really limited, and most of the fats will be in the form of triacyl glycerols, and same for oil as well. So they are non-polar in nature, and they are water insoluble, and they are triesters of glycerol. So they will have a glycerol, one glycerol molecule. Okay, glycerol, it's all, it means it's alcohol group. So it does have a 3 alcohol group, and these 3 alcohol group will react with 3 separate fatty acids with a carboxyl group and then form the ester linkage. So as you might know from chemistry lectures, the ester linkage is normally formed when an acid is reacting with alcohol. Okay, so here the alcohol groups are coming from the glycerol. So the alcohol groups are reacting with the carboxylic groups of 3 separate fatty acids and making this ester linkages. That's why they are called triacyl glycerols. Okay, or esterified fatty acids. So they will normally remove 3 water molecules during these reactions. So basically these are the energy reservoirs. So tags are energy reservoirs in animal, and they are most abundant form of lipids, and because of non-polar they are stored as anhydrous form. Okay, because there's no water molecule, they removed all the water molecule when they are making the ester linkages, so they are stored in anhydrous form, and therefore they can be stored in large quantity. But on the other hand, if you look at the glycogen, that's a glucose storage, that will be stored in hydrated form, so it will have twice the amount of water, therefore the storage level will be reduced. But here, because it's anhydrous, you can store more amount. So the fats produce 6 times more energy, and most of the energy will come from this triacyl glycerols. Okay, this is what will enable us to live up to 2 to 3 months in starvation. So without any food, we should be able to live up to 2 to 3 months just using our fats stored in our body. But if you look at glycogen storage in our body, that will last only for less than 1 day. Okay, so if you don't eat anything, if you are starving, your glycogen will run out within a day, but the fats will last for up to 2 to 3 days, 2 to 3 months to provide energy for us. Okay. So especially they are stored, tags are stored and synthesized in adipocytes. Okay, so adipocytes are the special fat cells in our body, and that's where most of the fat will be stored whenever we got excess amount. Okay, so for example, in abdomen region and the back. So this is where most of the fat will be stored. And if you look at the adipocytes, they are mostly or entirely filled with fat droplets. You can see here the large fat droplets sitting in the middle, and that's occupying most of the cell, and it's pushing all the cellular organelles towards the edges. So this is how large amount of the fats are getting stored in our body. So tags are mostly abundant in subcutaneous layer or abdominal cavity. As I said before, under the skin, there's layers of adipocytes with a large amount of triacylglycerols, and that's what acting as a thermal insulation in our body. And again, in abdominal cavity, and when we get older, or if you are eating too much, this fat keep on accumulating, and that's where they're getting stored. Okay, and the subcutaneous layer of tag is providing thermal insulation. Okay, I'll stop this here, and then we will move on to the next one. Sorry, the normal fat content in our body will be around 21% in men and 26% in women. Okay, I'll stop this video and continue in the next video. Thank you.